Here we go back. Aujourd'hui, today, we're going to talk about global change and mitigation aspects. Actually, we already did that this morning with Emma Sandrini, with the presentation that was fully private and fully interesting. And we will continue this morning with you, Roman, and we will finish with Mathieu, talking about ocean acidification with you, Roman, and with climate change effect on Arctic and thick communities with you, Mathieu. So, for this presentation, your jury members, your three jury members, Joanna Stochescu from Nature Dive, of course, you know Dynamian from Lab at Université Côte d'Azur, and online I have Leah Piacentini, PhD student at the University of Corsica, and also former student from RS class of 2020. Okay, and so I'm just a chair and not a jury member because I already had a link to your presentation 8 times. So here you go, and as Aldine just said, enjoy.
Okay, so you tell me when I can start.
You can start.
So good morning, everybody. Today I'm going to present you the work I've done for my Immersion project. So I had a chance to work at the Institute of the Sea in Villefranche-sur-Mer with my supervisor, Jérémy Carlo, on belting functioning in the face of global change. So global change is a threat to marine ecosystem at different levels and on different organisms. So our study is crucial to better understand ocean acidification and will help guiding future research priorities. This leads us to our study, our question: what are the effects of ocean acidification on benthic communities? To answer this question, I performed a meta-analysis. I focused on the benthic community, so the organisms that live at the bottom of the sea, and on natural analogs, but I will go further on it in the next slide. So our objectives are first to gather relevant studies and extract the data to create a database. So this was my main job during this 4-month internship. And then the second phase is to analyze the data and conclude on the effects of OA. So OA is ocean acidification. So I'm talking about ocean acidification, but what is it? Ocean acidification is a process where the pH decreases due to the absorption of carbon dioxide, the CO2, from the atmosphere. It results from the human activities, principally the combustion of fossil fuels like coal, but also the use of fertilizer, human activities in general. When CO2 is released into the atmosphere, a strong portion of it is absorbed by the ocean, leading to the formation of carbonic acid. This process decreases the pH of the seawater and make it more acidic. So what does it imply looking at marine life? Some species will be more vulnerable to low pH than others. It's the case for calcific species such as corals, sea urchins, some mollusks that need calcium carbonate to build their skeleton. So what is accompanied by the reduction of the pH is the decrease in the concentration of carbonate ions. So this is problematic for this kind of species. But ocean acidification has various effects. It can enhance the growth of some seagrass species, while it can decrease the growth of certain seagrass, but enhance the growth of certain kelp, for example. So more than a simple effect, it's the functioning of the ecosystem that are impacted. But the term function has to be defined, so it's the specific role an organism plays within an ecosystem. So it is important to understand this because it helps us to better understand how the organisms interact together and how they all contribute to the ecosystem functioning. So it can be the photosynthesis, the calcification, the growth, the biomass, etc. As I mentioned earlier, we're talking about natural analogs. The natural analogs are natural acidifying environments characterized by the pH that is lower than in normal condition. So it offers a natural experimental condition where it's not the case in laboratory because we have control parameter, and here you have how they can interact in their natural environment. So those are examples of natural analogs. And we performed a meta-analysis on the effect of the Benti communities, looking at these natural analogs. So the meta-analysis is a statistical method used in research to combine several independent studies on the same subject. So how did we perform this meta-analysis? So the first part was to gather the studies and create a database. So for this, we first created a hit list. So a hit list is a combination of several studies. So we took about 20 studies that were eligible articles on the subject. So we were sure that those articles corresponded to our research question. At the end, we have a percentage of correspondence between this hit list and the total amount of articles that we found. So then we define the query, which is the base of our analysis. So it's a set of different keywords that will help to select our articles. So we use the PECO method. So the P stands for population, so the population of interest, so the Benti communities, and also the geographical population, where you have natural analog in the words. Then the keywords on exposure, so low pH, low PCO2 acidification. So the C is for the comparison. So in our case, it would be the normal condition, but we did not take into account keywords for this one. And then the outcome. So what is measurable, like what is useful to measure an effect? So in our case, it's related to the function. So it would be calcification, biomass, growth rate, etc. So then we launched this query on R, and we got about 2,334 articles. So that's a lot. So we had to select them to because not all of them were relevant for our study. Some were not talking about natural analogs, or were not talking about the outcomes that we're looking for. So we had to do first a title and abstract screening. So we excluded some article, then a full text screening, and then we kept the articles with the data that were relevant. So now we extracted this data to create the database. For this, we divided our study into 8 processes. So those are the following processes that we took into account. And we use the website Webplot Digitizer, where you can directly extract the data. So you can have a graph and put the Y and X axis, and you extract directly the data when you don't have, for example, the tables available in the articles. So now the second part is the meta-analysis, so the analysis of the results. So we homogenize the data because for several studies, you don't have the same units in the same outcome. So we needed them to be homogenized to be comparable. And then we performed forest plot analysis, but I will explain it in the results. So what did we find? This is a map of the studies that were found. So we have a lot of studies in Papua New Guinea, Japan, and Italy, and a little bit less in Mexico, Spain, and Australia. And in gray, we did not find any articles on this area. Now, I decided to focus on the most studied outcomes in the articles. So we found about 22 articles that were talking about photosynthesis. And this is the number of observations. So for photosynthesis, we have a lot of different observations for the studies. So the respiration has the most observations, and we have the calcification, which were highly represented in 19 papers, which lead us to the most groups represented. So we have the corals that were highly represented in terms of observation in all the studies. Then we have the community. So the community were not really defined specifically. It was like the benthic community in general. So they didn't separate the algae, the seagrass, or the invertebrates. Then we have the algae and the seagrass, and the less represented here is the kelp. So now I'm passing to the results of the second part of the analysis. So first, this graph is just for you to understand what is a forest plot. So it's based on the effect size, which quantify the strength of an effect or a relationship. So on the left, you have the significant negatively impact. Then significant positive impact. Here in gray, you have no significance. And in black, it's when the data were not enough to be concluding to have an effect. So with the help of my supervisor that gave me, so we understand the functioning of this forest plots, I had to analyze the results. So we have a significant negative impact of ocean acidification or other biomass. So the others here is the bryozoans and sponges. Then we have a significant negative impact of ocean acidification on coral calcification, sorry. And then we have a significant negative impact on algal community, as well as negative impact on seagrass and algae growth. And the only positive impact of OA that we found was on invertebrate growth. So I decided to show you the only significant results because the other graph did not show. So for example, for the respiration, we did not show any significant results, or the data were not enough to conclude. But how can we interpret this data? So first, it's difficult to conclude on the negative impact on other biomass because it's very specified to these sponges and these bryozoans. So I think that other, it's hard to make a conclusion because it's the category. We wanted to include this data, but we didn't know how to separate the categories. So that's one aspect that is to be considered. Then we show negative impact on algae complexity, which has already been assessed in some studies, but there is a lack of studies on algal complexity. So that's why we can discuss about this negative impact. And also there is different methods to assess the complexity. So it can be a reason why we have these results. Then we see the negative impact on sea growth and algae, which was a little bit surprising because when you look at literature, you have a lot of seagrass growth improvements when phase to low PCO2, and also for some algae. But the problem is here that we englobe all the algae together while you have different type of algae. So you can have macroalgae, turf algae, calcifying algae. And so calcifying algae are more vulnerable to low pH than turf algae that usually thrives when you have ocean acidification. So that's a problem that we could separate the different categories to have more accurate data. And then, of course, the negative impact on coral calcification that aligns with global literature. We found it's in a lot of paper, which can also be a reason why we have a lot of corals observation in the article and why you have a lot of studies in the 62 studies. We had 19 studies on coral. And then the surprising impact was on the invertebrate growth. So usually in literature, you found that invertebrate growth decrease with low pH, but one of the reasons we can hypothesize that they can improve. So what is important to know, it's some species of kelp and seagrass will increase the pH locally. So they have the capabilities to increase the pH locally in acidified environment. So we could imagine that the invertebrates that lives close to these species could benefit from this increase locally while they are in a bigger, like a low pH environment. So our study have some limitation, but perspective. So like I said, the choice of taxonomic group in our meta-analysis, we only divided into algae, seagrass, but we could specify the type of algae, like turf algae, macroalgae, calcifying algae, and not only species. Also for the invertebrates, because you have calcifying invertebrates and non-calcifying invertebrates, and the selection of articles. So in our meta-analysis, we selected articles that were from Web of Science, and we could improve the query and the codes to make it look for articles in other research engines such as Google Scholar and other type of research database. Also, which could be interesting, is to compare this meta-analysis did on natural analog to a meta-analysis did on laboratory experiments. So it could be a reason why we have different results, because in laboratory you have controlled parameters, which is not the case for our natural analogs analysis. So as a conclusion, this study gave us some effects on the key ecological function. It highlights the negative impact on coral calcification while giving us an enhance in vertebrates growth. So this is important to consider the study on natural analogs because it gives real condition of acidification, and it emphasizes the need for conservation measure. So for this presentation, I would like to thank my supervisor, Jérémy Carlo and Nuria Textillo, and also the Maris team, Crystal and Christophe.
So let's shift opening the floor for the questions. So do you want to start, Jonas, and we can go to you, Leah, and we'll finish with you.
Okay, well done for the presentation. I find it super interesting, and it's really hard work that you did. I was just a bit confused about using the word invertebrates. You say there is a positive impact on invertebrate growth. So do you include corals here? Because corals are also invertebrates.
Corals were separate for the invertebrates. So that was more so the mollusks.
Okay.
Yeah.
And when I first read your presentation at home, I had this question of how do you, if, for example, it can be the case that the studies have actually, what happens when you introduce into your data extraction studies that are contradictory, for example. But then I think I understood that you already answered the question in your on the slide where you explain the forest plot. So it can be the case that, for example, contradictory studies can have repercussion on the black spot in the not.
So the black spot, it's like the data were not sufficient to conclude on the significant or not significant, but yeah, the negative or the non-significant can be balanced between, for example, the example of algae. If we have calcifying algae response and turf algae response, then it can maybe balance it to make it non-significant as this thing.
Thank you. I understood better now. So well done for presenting that. What was the greatest challenge in this study for you?
I think so. The results definitely to understand how to homogenize the results. So I had the chance to have the help of Jérémy. But also, there was a really long work to go through all the articles, especially because at the beginning, when you see that there is more than 2,000 articles, you're a little bit scared. But then it's easy. You have you take the habits and for information, and then you see the information at the end. But that was at the beginning one of the greatest challenge.
Well done. Thank you.
Yeah.
Hi. Thanks for your presentation. Also very clear. So, as you already told on the discussion and perspectives part, there is a wide diversity of organisms in your 3 or 4 categories. And do you think that if you separate it in smaller categories, like for invertebrates, for example, there is a very wide variety of invertebrates. Do you think that you could obtain results? Because I see in the calcification forest plot that for the invertebrates, for example, there is no significant impact of the ocean acidification, but it's slightly towards the negative part.
Mhm.
And then we see that for the growth is significantly positive.
Mhm.
So can you explain that?
Yeah.
Give
So I
Yes.
think it will really depend on the type of invertebrate we chose and the invertebrate that we had in the articles, because it's possible that we excluded some articles that were more focusing on certain invertebrates. So I think in this case, yeah, it's hard because it's like the thing with the algae, because you have some invertebrate. If you look at literature, you have some invertebrate that are vulnerable to classification, but not all invertebrates are classifying species. So it's hard to. That's why we could definitely improve the group specification and maybe put invertebrates and then separate into several groups.
And do you think that if you separate in several groups, you will have enough data to have some results?
So that's the thing. It's because we have so many groups, like we are taking into account the corals, the seagrass, etc. So I think to have more accurate data and more precise data, it would be interesting to do a meta-analysis on only invertebrates, so we can gather more studies, because also it depends on the query at the beginning. So the keywords that you put. So maybe we could improve the query and the keywords, so it can look for more keywords in. So the keywords were in the abstract, title, and the keywords in the article. So we only put. I have the. So this is the query. So it's a long. So this is the query, the population of interest. So we. So we put invertebrates here, sorry, and we could specify that's what I did at first. Like I separate like gastropod, like every type of invertebrates. Then it was too large and we had too many articles. So when you have like, for example, when you launch the query and you have 10,000 articles, it's not possible to start a meta-analysis with this. So you have to reduce the amount of keywords to only have like the really relevant studies. But that's why we can miss some paper.
Okay. And so it's a very wide question, but what is the next step then? What will you do next?
So next, so the next step would be to interpret this data and maybe put combining this data with in situ experiments. So that's the case that what they already been done in Ischia in Italy. So where there is natural analogs, and they study the photosynthesis, the respiration, to complete the data that we found and to making it more relevant.
Okay, thank you. That's all for me.
Okay, Edine?
So thank you for this nice presentation. So to come back on the topic, so you talked about climate change, you talked about ocean acidification. Can you explain me the link
between climate change and ocean acidification? Well, it's as I said, the human activities that are the main, like the main, how do you say, the consequence of the human activities are the global change, and so this is linked to the absorption of the excess of CO2 that we have in the atmosphere that is absorbed, and this will lead to the ocean acidification.
Thanks. So what is nowadays the importance of the work you perform?
This work. So it's important because ocean acidification is we have a lot of studies on global warming, and it is known that it impacts marine life. And so it's important so we can have more insight of what it actually does on the marine life. So we can do more studies, and we can maybe implement a conservation measure to.
Yes, but you use a specific methodology
Mhm.
to tackle this question. So why it is important to do the job you perform now with the methodology you perform regarding this field?
I would say to have like a global visualization of the effect, because if you know that at this or in this area of the world you have this effect, it has a lower impact than if you say that it's like a bigger impact and it affects all the area in the world combining.
So can you go back on your page 6, your slide 6, and the point 3? Yes.
Yes.
Your third point. So can you comment on that, please? I didn't.
So that's the link between so data and organism to the okay. So that's the link with the functioning. So it's if ocean acidification will affect the type of organism, but it will not necessarily lead to the dispersion of this organism. It will lead to like an impact in their function. So the total ecosystem will function differently. So that's it's the link between the function and the ecosystem.
Okay, thank you. So for your methodology, so to select your paper of interest,
Mhm.
Can you come back on the criteria you use to extract this?
The query.
Yeah, the query. So
on the slide or at the annex.
well, which criteria did you use to select the paper you will work with?
So I'll go back on the query. So with the query, so we'll look for, so in each, so how do you say? So when you have an article, you have the title, the abstract, and the keywords. And to select the article, we needed to find in these three, at least one of each. So we could have a study on the seagrass in the Mediterranean, and they were talking about biomass. So it's a global thing, and yeah, that's how we proceed. So we had to find at least one of each keywords in each well, either title, abstract, or
Okay,
the
so
keywords.
it's at least. It's not a combination of a certain number of criteria.
No. It's at least one of them. That's why sometimes we had that's why we had a lot of article. It's because for the exposure we put acid.
Mhm.
So it could be the little star it could be acidic, acidified. It's just the beginning. So you can have we're acidic. So that's why we had a lot of paper on molecules that didn't talk about ocean acidification. It was just about the acid in some gastropod or.
Okay. Do I have more time?
Yeah.
Okay. Can you come back on the PECO method? PECO. I totally don't know. So can you explain it, please?
Yeah. So this represents the query. So we had to define the population, so the type of organism. So we divided into population of interest, so the benthic community. So the first one, the corals, benthic, seagrass, etc. Then the second part was the geographic population. So where do we find the natural analogs?
Okay, it's what you call exposure.
No, this is
I know the
population.
population.
Then exposure is what is it exposed to. So in the title or abstract, we can have not necessarily ocean acidification, but how they react faced to a low pH or low PCO2. So that's what was the exposure. So all the keywords related to ocean acidification. And this we didn't take into account because it's not like we don't care, but it's not necessary to have the comparison in the title or in the abstract because we imagine that it will be compared to to a normal pH or neutral pH. So it's the PICO method used this for, but in our analysis we didn't put any keywords on no pH because it would reduce the amount of study that we will have at the end.
Okay, but it can be used to refine the resolution of your study and to focus on.
Yeah.
deeper
You could also add it, but not so put it like a supplementary.
Okay.
Because we had to have at least one of each,
Mhm.
so this one would be like
Okay,
additional.
you want to start it broad. Okay, thanks. When you are talking about the data homogenization, what are you calling homogenization in the context of your study?
So, for example, for classification, you have a lot of in a study they will measure it in a specific units, but in another study they will use another unit. So, for example, it can be basic, like from square per year to gram per.
Okay.
But sometimes it's harder because they use the mole, like a mole of C or mole of O2
Mhm.
in gram per meter per blah blah blah, and then we have to homogenize all of them. So that's one of my job, particularly. That's so I homogenize the data, and then I needed some help because some units are very complicated to.
Then related on that, so when you are saying that there is a negative impact, positive impact on invertebrate growth,
Mhm.
Which kind of life stages do you have in the paper? Because what are you calling growth? And
That's the one of the limitation. It's because we don't really take into account the different stages, which are different. We know in literature you have different effects depending on the stage of the organism.
because often they are looking, the development, submarine development, or for the growth, it's more about coral growth. But for an invertebrate growth, what are they calling invertebrate growth? So, okay.
This could explain also a positive impact because it depends on the study. Maybe like I don't remember honestly on the 62 study how many were on the invertebrate, but it can definitely be if we had other study at different stage, maybe the results would be different.
I have a last question. Okay. So if you can come back on your map, can you just detail a little bit more and explain what this map is showing?
So at the end, we kept 62 studies.
Okay.
So these 62 studies have been studied in natural analogs. So most of them happened in Papua New Guinea, Japan, and Italy, where you have a lot of natural analogs, CO2 vents mostly. So this represented in orange, and like when you're in blue, it's that you have less study about it, that have been done in this area. And in gray, you don't have any studies that have been done there.
But the US and the entire Europe, it's quite super surprising. So can you comment on that?
It's because it's based on the natural analog. So you have most of the study. So you have a pruning area, rainfall area that can act as a natural acidified environment, but most of them are CO2 vents. And most of the CO2 vents you can find in the world are in this area. I have a map so they have done an analysis on CO2 vents, and this is the map. So these are where the vent sites are mostly. So you have a lot of in Italy, in Papua New Guinea, and in Japan.
So in your map, in your first map, Italy is colored or not?
Yeah.
Okay. Sorry.
There is Panarea.
Yeah.
It's with a lot of CO2 vents there,
Okay.
and also in Mexico.
Yeah.
But you don't have natural vents.
Yes.
Well, that is known, for example, in Africa.
Okay. Thank you so much.
Okay, I think we're good. Roman, question again. It's your time, Mathieu. Ah bah oui.
Demain.
Tomorrow, yes.
Yesterday.
Okay. Quick for you. For once, I will take the time to change the name before you start. Erika, tu dis bravo. Non, je disais Erika, tu dis bravo. Okay. Do I ask you, how are you?
Hi.
Mathieu, so your turn. 15 minutes and then we will discuss your presentation. Yes, okay, go ahead.
Okay, so hello everyone. Just can I put my
Ah oui, bien sûr.
up? So hello everyone, I'm Mathieu. I'm here to present you my immersion project that I did at the IMEV, so l'Institut de la mer de Villefranche-sur-Mer, about the effects of climate change on Arctic-Benthic communities. So for a little bit of context, today global warming impacts both inland and marine ecosystems. Factors such as pH, turbidity, nutrient availability, and temperatures are really improving. And these factors can have various effects on the marine ecosystems. What you can see on this map is the global repartition of kelps genera in the world. The main density is located in the polar, subpolar, and temperate waters, and we can't find any kelp species in the tropics. Today, the studies of ecosystems are mandatory as they act as source of food, nurseries, and living areas for many organisms. They are also important in the carbon cycle as they act for carbon sequestration when they are doing respiration and photosynthesis, but also when they are dying and releasing the carbon in the sediments. So now we want to determine what are the effects of climate change on Arctic-Benthic communities, focusing on the kelp forest. So our study area takes place in Svalbard, so in north part of Norway, with the Kongsfjorden that represent our fjord of interest, as there is the Ny-Ålesund scientific station. This station have all the abilities with boats, lab to study the effects. Also, there is the Roe Glacier that lost 2 kilometers from one century and 1 kilometer from only 30 years. That's an interesting site because the different sites can have variations about the impact of glaciers. So we have decided to choose two specific sites. The first one is Ansniset. It's located at the middle of the fjord. It's close to glaciers in a sheltered area. So there we can find an high turbidity. The second one is Kongsfjorden. It's located at the mouth to the beginning of the fjord. It's really much more exposed to wind and waves, but less exposed to the melting glaciers. So Kongsfjorden will be our control site, and Ansniset will be the site representative for the future. So to do our analysis, we have several aims and objectives. The first one is to do the first net ecosystem production that I will explain to you after, in situ in the Kongsfjorden and globally in Svalbard, focusing on three most abundant kelp species there. So Saccharina latissima, Laminaria digitata, and Aloria esculenta. The second goal is to determine the global shape of each community. So by observation, thanks to an ROV. And the last one is to adapt a protocol developed by Takeshita and Al in 2016 on coral reef, adaptable to kelps. So here's our two methods. The first one is the remotely operated underwater vehicle. So that's basically observation of the shape of the communities, thanks to a camera, and when we can make films and then extract images. The second one is the net ecosystem production. So the net ecosystem production is a rate that indicates the dependence of external sources of carbon versus what an ecosystem can produce on its own. So it takes into account the respiration and the photosynthesis. And basically, if a rate is positive, it means that the community retains more carbon that it releases. So our first method is the ROV transect. We tested different methods. The first one was to just have 40 meters of cable of the ROV and try to do a straight line. Also, we tested directly to attach a line to the ROV, but many problems appear for linked to the current, for example, that move the ROV, so we can't really have the same sampling method each time. So that was the easiest one. It's a 40-meter transect. The drone is piloted by the boat. We are running one way, and we repeat the method 3 times at each site. What we obtain is basically pictures like that. We can see there, it's small, it's linked to turbidity. There is 2 small dots. These dots are separated for 7 centimeters. That 2 lasers that installed on the ROV. And then after it helps us to create a scale to analyze the size and the whole front area of each scale. Concerning our NEP, to do the calculation of the net ecosystem production, we need several sensors that we attach on the structure. So the first sensor is the O2. The O2 sensor is directly linked to a pumping system. We are working on a gradient flux approach, so difference of the gradient about the O2 concentration in the water column. So there is the 4 different heights. The first one is in the kelp forest, and the last one is above kelp forest. To run this, we need a computer that will change the pumping heights every 4 minutes and link to a battery that allows us to retrieve data for a few years to a few days. The last instrument is an ADCP, an acoustic Doppler current profiler. That's an instrument that's sending pings, some things in the water column, and it's creating cells about different heights, and it can determine the speed of particles in these cells. So here's our NNP calculation. The first one is to take into account the friction velocity, so the movement of fluid in the water columns. So basically, it's the current velocity, but also the von Karman constant, so the logarithmic velocity, the turbulent fluids. The height above the bentos where we placed our structure. Also the particle displacement, so the difference is particle that we aim to target, and also the roughness scale. Then we can calculate directly our NLP. As we said, there is a gradient fluid approach. So we take the dissolved oxygen from the top, the dissolved oxygen in the kelp forest. And also we have to determine at which height the pumping system is. It's taking into account the water density and the U star, so the velocity that we calculated just before. So let's have a look at our results. The first one is about the number of individuals that you can see. There is 42.2% more individual at Ansniset, so the site close to the glaciers. And we have globally the same pattern about the abundance of the species on both sides, with a dominance of Laminaria digitata. Then, if we have a look on individuals, the average front area is also bigger for both for the 3 species in both sites. And the most impacted species is Alaria esculenta. So 45% more front area for Alaria esculenta in Ansniset, 7% more for Saccharina latissima, and 3.8% more for Laminaria digitata. Our NEP results for both sides. So we have seen that there is more individuals in Ansniset. Individuals are bigger, but the net ecosystem production is lower at Ansniset. And so these are the results for both sides. One for 40 hours deployment and one for 60 hours deployment. At least it's a positive net ecosystem production. So that means that the forest retains more carbon that is being released. And this rate is in minimal carbon per meter square per hour. So what we have seen, it's first of all, for the remotely operated vehicle, when we did our observation, we have seen way more individuals in Ansniset, way more bigger individuals in Ansniset. And that can be linked to several factors. The first factor that we can discuss is about the current. The current can degrade or detach some algae from the bentos. And this can be linked to the shape of each individuals. For example, we have seen that 80% of the population is Laminaria digitata, and this species have a morphology of 10 blades, allowing the current passing through. And and the two other species are in one blade, and they are thicker. So they are taking way more current, and they are way more impacted by this. And we could also see that when we had the observation. In Sconfergoniset, the whole population was spread into patches, and in Ansniset, the density was way more. So concerning our NEP, we had an NEP, a positive NEP, but close to zero. And what we have seen in the literature is that this range there is an EV range of NEP from -4.7 to 4.6. And this can be linked to the temporality, the sites, the weather. Many factors can impact this. And also the method used. In some projects, the method used have been discussed in the literature, and scientists are not agreeing on the same methods. And on average, selecting only the good methods and the good temporality, the net ecosystem production is around 2.9. So positive one, way more that we have. And so it can be directly linked to the turbidity, so the climate change that directly impacted our glaciers. The turbidity can impact the productivity of kelp species from 97%. And particle risk by glaciers can be deposits on the leaves of kelps, and occurring both in the water column, the light to enter, but also the light to enter the individuals. We have several limitations on site for the observations. First of all, the number of sites and the number of transects we have been able to do because of the weather that not allows to go on to do our observation each day. And also a high turbidity that sometimes when you retrieve data, you can't see your scale, etc. For our net ecosystem production, same. It's cool. We had first of all targeted 3 different sites. At least we could do only 2, and the techniques are really different in the literature. That's sometimes complicated to compare our data. Our conclusion: we have been able to carry out our first NEP characterization in the Kongsbergen and globally in the region of Svalbard. And we have been able to see the dynamic of these kelps. We have been able also to prove the impact of the turbidity and the current on these different communities, and coupling our observation method with the ROV with the NEP allows us to better understand the dynamic of the kelps between the sites inside the fjord. I would like to thank my supervisor Steve Como and the whole Orca team, so Pierre Ruti and Samir Aliwan, and also the people I spent time with in Arctic, so Kay Miller and the whole Spanish team that working on red algae there. Thank you.
Thank you, Mathieu. And we will continue shifting the start of questions with you, Leah. Are you ready? Your microphone is mute. That won't be funny otherwise. Yes, I confirm. You're mute.
Is that okay?
Yeah, oui.
Okay, perfect. Thank you for your presentation. Very interesting. I'm not sure, maybe I missed the information, but how many transects did you do for each site, and at which frequency?
It was 3 transects at each site. I perhaps forgot to mention it. Yeah.
Okay. And I saw that you had some graphics, and maybe on the slide 18, you say there is a 45% more, etc., etc. Have you done any statistical analysis on that?
No, it's globally just an average. It's an average that I did on the observations on each animals. And for each species, we did an average of the whole area of the fronts for each individual. And
don't you think it will be interesting to do some statistical analysis?
Yeah, for sure. But I had to focus on a few datas. Even, for example, we can add during our analysis of NP, compare with SPAR data, so with a light logger, etc. We used one in the article, for example, but sometimes we are losing data, or the parameters are not well implemented in the light logger. So it's taking, for example, for one site each 10 minutes, in the other site each 1 minute. So sometimes I struggle just analyzing each part of our observation. So I just focus on the average front area for
Okay.
Yeah, for sure.
Yes, okay. And in a perfect world where you have all the data you wanted and every parameters were accurate, acquis, what would be the analysis that you would do on those data?
First of all, it will be the characterization of the impact of the current of each individual. Because we saw detached animals, individual. We saw degraded individuals, but with observation that's complicated. But yeah, as we said, if we have perfect data analysis, we can have more robust analysis to do. But with only few pictures that are enable us to do it, that was not enough, I think.
Okay. That's.
Yeah, we can come back to you if you want.
Yes.
After.
Okay.
Yeah.
For now, it's all I have.
What did I say? Fiona. Yes. Alex, go ahead.
Great. Since we're also on this slide, I was curious, first of all, why, how does the number of individuals that you observed with a ROV, how does that respond to your research question regarding the impact of climate change? I mean, the number, can it be like, is it super related to the climate change itself, or can it be, for example, the difference in number of individuals related also to the substrate, or like to
It was, first of all, to see if the turbidity can impact the whole shape of the community. So I wanted to know if there is less individual where there is high turbidity, for example, if the nutrients availability there is more nutrient available at Anthony-set, but the mixing is less. So I wanted to know if in Conferganisset without this turbidity, there is more individuals, etc. And what we've seen is that in the site where the turbidity is high and there is more nutrients, etc., there is more individual and they are bigger. So that's probably linked to the glaciers. Even if there is this turbidity, they are still able to grow and there is more animals. So the current seems that are not directly from the global warming. The current is not linked to the global warming. And this is the main factor that impacts the shape of the community and not the turbidity in this case.
Okay, because can you say for sure that, okay, it's turbidity that influences the number of individuals, or can it be also the dynamics of the ecosystem itself? Because there are other species also, I imagine that there are other species that have
that's the problematic of for example, the NEP and the number of individuals observed, etc. For example, the NEP, when you calculate it, it's taking the whole community, so it's taking into account the green algae, the red algae, etc. So for this, it's the same. Sometimes, like I write laminar data, laminar nucleus, we can't sometimes really differentiate the individuals, etc. And so we don't know if this species is more adaptable to this site. So yeah, there is few limitation like that to better understand the shape of the community. I don't know if it's what you're thinking about.
Yes. And if you were, for example, to eliminate, let's say, for whatever reason, you would not have the possibility to use the ROV. If you would eliminate it from the experiment, would you still be able to reach some conclusion just with the NEP method?
That's complicated because with the NEP method, we only have the production and what's releasing. So we don't know, for example, why it's we just have the answer of it's releasing it's releasing more carbon in Ansniset, but we don't know the shape of the population, etc. So I think it's mandatory now to check the shape of the community before, and that's why it's discussed in the literacy now that the methods are really different between sites and studies.
Cool. And would it be possible to use the images that you filmed to create photogrammetry? Have you thought about that? And would photogrammetry serve to your study?
The photogrammetry, so it's analyze the colors.
No, using the images you filmed with the ROV to create a 3D model.
The 3D model.
Yeah.
That's as I said, with the high turbidity, that's complicated to see more than most of the time 50 meters. So to really know where the drone is, we have to couple this analysis with perhaps already shaped data or something like that, because with just the ROV, that's really complicated due to turbidity.
Thank you. Well done. And what was the best lesson that you came with from the Arctic?
The best lesson?
Did you have a best lesson for you?
The adaptability of in situ measurements. Sometimes we have to really adapt methods. We adapt all the plans because of the weather. Because one time we retrieved data for Ansniset and our structure totally flipped due to an iceberg that hit the structure. So we lose 4 or 5 days of data, something like that. Wow. So sometimes, we have to really adapt the technique.
Thank you, and well done for the presentation.
Thank you.
So, Adine.
Thank you, Mathieu. So, your title, it's about, and your question, it's about benthic communities in general. And then you focus on few species.
Yeah.
So with the methodology you are using, what is the resolution of the species you can go? And I'm really surprised that there is only 3.
There is more species, but that's the 3 most abundant.
Yeah.
And we say benthic communities as we are looking on net ecosystem production, and we can't separate the species. So we know that we're gonna have some interference by green algae, red algae, etc. So that's why we are talking about the Arctic benthic communities, as we can target really species. But to go further on that, the incubation chambers will be a great idea to know exactly the impact of the net productivity of each species, and then better characterize. Yeah, but also focus after on green algae, red algae, etc. to combine all the data.
But in terms of the methodology you use with a lot of examples, so what is at the macro scale, what the smallest organism you can visualize? This methodology.
I think for me it's the red algae. There is a few general red algae. I spent time with the Spanish team there where they targeted the red algae, and on video we can see it, but that's really complicated to have the moment where we can see the scale, and at the same moment the individual that we target. And as they are more rare, we need the perfect moment to.
Are they more rare or they are covered by the other one?
Both, yeah. because kelp species can be more than 1 meters and etc. So
They have the same ecosystem.
exactly.
Okay.
On rocks.
If you have to come back on the goal of your study, so what was the goal of your study?
This one's the aims or the main goal of my scientific question.
So now that you perform all this work, so when if you have to mention your main goal, so what they will be?
It was the first net ecosystem production of Svalbard, of the Benti community in Svalbard. And that's a thing that is where more developed in the world now. And that's something to be improved. Protocols like Takeshita developed, etc., needs way more studies to better target the importance of each parameters in their protocols. And using these protocols based on corals on kelps allows us to better understand what we have to take into account for kelps that it didn't take into account for corals, for example. And that will lead, I think, in few years to a strong method that everyone can use for kelps, everyone can use for seagrass, etc. So for me, that was the main goal, to really adapt this method and have relevant data on that.
Okay, thanks. Then I think it's fine for me. Yeah, that's fine. Thank you so much.
I'm not asking questions, but I'm curious. You're talking about ecosystems that a few people on Earth have the chance to see, even with pictures. Do you have pictures or videos for us?
I have pictures every now, right now.
Yeah.
Well, the one that I use, it's at the end and at the start. That's my picture. I didn't mention it, but that's my picture.
Yeah, but of the bottom,
Underwater.
on the underwater.
Underwater, we have this picture on the drone. That's the best one that to show what we saw. That's
It's not to organize things.
really, that's really like that.
You're experts.
Just it's a simple advice, and it's for everyone, that not forget that you're in marine biology as well, and that you will like to see pictures of real animals,
Oh, okay.
and we like to see Latin names in Italic also. But yeah, for everyone, keep that in mind for the future when you come back after your internships. We like to see pictures to see in real what you're talking about, and especially for you because you worked with pictures or videos. We would like to know, yeah, what was the real part of it. So even if you pass quickly, it's important.
Can I ask the last question? But
No.
okay, it's not
You want
entry
to
data.
read
And, yeah. So now you have all this data and you test all these different methodologies. So what is missing for you now? What you would love to do to really complete this data or to improve this methodology?
Okay, first of all, to complete the data, for me, it's adding a site with a strong current like Kongfagan set.
Okay.
To know the dispersion, because for the NLP calculation, we use the dispersion. So it could be really interesting to have a strong impact from glaciers like Anthony said, but also a strong impact of the current. Also, it could be great to do it. I know that's complicated, but do it each year, taking into account consider the storm events, the global weather, the time melting, because the polar summer is not starting at the same day each year. So it could be great to determine the dynamic of the glacier, and also to compare with the dynamic of the community at the same time.
All good. Thank you, Mathieu.
Thank you so much.
So we will come back this afternoon with Anaïs at 1:15. Anaïs, you will be the only presentation with live, because after that we'll have Maya, and you're all kindly invited in this room for support Maya. And we'll finish with Luz and Esther. We'll have the weird experience to be alone with us in this room.